Do we have any remyelination therapies? So nothing licensed, but lots in development. And so that's one thing that's happened over the last 10 to 20 years is the unpacking of biology around the repair mechanisms that are needed to stimulate remyelination in multiple sclerosis. And we now have quite a few biomarkers for picking up remyelination in people with MS. We can actually use electrical tests. So when you have an inflammatory lesion, say in the optic nerve, optic neuritis, and it recovers, you get a thing called a visually evoked potential, and you get a P100, positive wave 100. 100 refers to 100 milliseconds. And when somebody recovers from optic neuritis, that's often 120 milliseconds, 20 milliseconds delayed. And you can now actually use that to try and improve the conduction. So that's one way of doing it. Another way of doing it is using MRI techniques. So around individual lesions, you can actually measure the myelin content using a technique called MTR, for example. And there's a thing called myelin water. And so there are a lot of now techniques that have been available for us to use as biomarkers to test remyelination. And one of the first drugs to come through was this drug called oportunumab. It blocks a molecule called LINGO-1, which is an inhibitor of remyelination. And that was the first drug to go through the development pipeline and was shown to improve conduction in the optic nerve model, in optic neuritis. And that's now gone into a phase 2 study where a subgroup of patients was shown to respond to it. And it's now in a phase 2b/3a trial to see if we can improve outcomes in people with established MS that have disability. So we're beginning to see the emergence of remyelination treatments. One drug that's also that's been tested right now in phase 3 is high-dose biotin. So biotin, as you know, is a vitamin. That's a cofactor for many enzymes. And there was an observation that in a biotin-responsive leukodystrophy, white matter disease, some people with multiple sclerosis were misclassified as having this leukodystrophy, and they were given high-dose biotin and improved. And biotin is probably got two modes of action in relation to MS. One of them is to improve energetics along axons, and the other one is to possibly stimulate myelination. And so this is another remyelination agent that's currently in trial in advanced MS, both in primary and secondary progressive MS, where the primary outcome is to try and improve disability. And we should get the results of that within the next 6 to 12 months. And that would be a very interesting add-on. I would not think that high-dose biotin will be used as a monotherapy. It'll be added on top of existing treatments. So we are seeing movement into neurorestoration trials. Neuroprotection. So we have had a flurry of trials looking at individual compounds as neuroprotective agents. The one that we've been involved in is looking at sodium channel blockade. We've used the anticonvulsant phenytoin. And we were able to show that in optic neuritis, if you go in with phenytoin very quickly, within the first 2 weeks of presentation, and you load up patients for 3 months on phenytoin, that you can actually protect the retinal nerve fiber layer. You can reduce the dropout in terms of nerve cell loss by about 30%. Now, the trial we did was too small to see a clinical benefit, but we need to take the sodium channel blockade hypothesis forward and now put it into a larger number of patients to see if we can improve outcomes. But again, it's not going to be monotherapy. Any neuroprotection has to be on top of anti-inflammatories, because there's no point in trying to put out a fire if new fires are starting. So the idea with neuroprotection is you have to then get on top of the inflammation and hopefully protect the damaged axons going forward. And there are a large number of compounds. We've got too many compounds to test, to be honest with you, because our current strategy for neuroprotection trials is quite long and laborious. And we probably need a much better phase 2 paradigm to screen out the best compounds going forward. And that's why we're trying to set up this platform in the United Kingdom where we would have like an adaptive design. We would have a placebo arm, and then what we'd do is we'd add new arms to test new neuroprotective agents going forward to see if we can have a platform for drug discovery. What about drugs that restore function? Difficult question. There are lots of reasons why we can restore function in MS. Forget about biological therapies that restore function. If we just stop this disease early, most people have, a significant proportion of people, up to 50% will have recovery of function. So I actually think the brain and the spinal cord are geared up for recovery of function if we treat this disease earlier. If you treat this disease late, after there's lots of neuroaxonal loss, the ability to recover function disappears. Another thing that we're not doing is we're not using what's been learned from other fields in neuroscience, like the spinal cord injury. To recover function in spinal cord injury, you put these people through quite aggressive rehabilitation programs, and you use exercise and repetition to improve function. And I think we in the MS space are going to have to learn, what can we do, learning from animal models and other areas of neuroscience, when we just start designing our neurorestoration trials, proper targeted, focused rehab looking at that specific disability. I think we've got a lot to learn still. But there are some really interesting compounds going into trial in MS. One of them is a monoclonal antibody that targets RGMA. That's the repulsive guidance molecule A. So there's a whole biological pathway there. It's an inhibitor of growth cone formation on synaptogenesis. So by blocking this pathway, you encourage axonal sprouting, you encourage synaptogenesis. And the basic science in the spinal cord injury field is remarkable with this compound. And it's right now in phase 2 in multiple sclerosis. And we're quite excited about the potential of this new biology being unpacked and being a treatment target to improve function in multiple sclerosis.